Hi, my name is Michael, and today we're going to be learning about muscle contraction and rigor mortis. First, let's go over the structures involved. Myofibril is a cylindrical organelle running the length of the muscle fiber containing actin and myosin filaments. Sarcomere is the functional unit of the myofibril divided into I, A, and H bands. Actin is a thin contractile protein filament containing active or binding sites. Myosin is a thick contractile protein filament with protrusions known as myosin heads. Tropomyosin is an actin binding protein which regulates muscle contraction. Troponin is a complex of 3 proteins attached to tropomyosin. Here are an example of a relaxed muscle, a contracted muscle, and a muscle in rigor. Now let's see how these work. Nerve impulse at axon terminal triggers release of acetylcholine. It diffuses across cleft, binds to receptors of motor end plate, triggering action potential. Acetylcholine esterase destroys acetylcholine, so another muscle action potential cannot happen until more acetylcholine is released. Action potential travels along T-tubule, opening calcium release channels in the sarcoplasmic reticulum. Calcium ions flow into the sarcoplasm. Calcium binds to troponin on the thin filament, exposing myosin binding site. Power strokes use the ATP myosin heads bind to actin, swivel and release, so thin filaments are pulled toward the center. Boom, contraction. Then calcium release channels close, and active transport pumps use ATP to restore low level of calcium ions in the sarcoplasm. The troponin tropomyosin complex slides back, again blocking the binding sites. The calcium triggers the exposure of the binding sites on the actin. The myosin binds to the actin. Cross-bridge power stroke slides the filaments to the ATP binds with the cross bridge, disconnecting it from the actin. There you go. So we've gone through the steps of the sliding filament theory. So now let's go through the rigor mortis steps. Step 1. Upon death, the calcium ion pumps of cells stop functioning due to lack of ATP circulation. Step 2. The calcium ion diffuses into the muscle fibers. Step 3. Active sites activate, causing cross bridges to form, locking actin and myosin together. Step 4. The muscle doesn't actually contract because there is no ATP being produced to fuel the pivot of the myosin heads. Step 5. Rigor mortis lasts 15 to 25 hours until lysosomes burst, destroying both actin and myosin. Here are my sources. Hope you enjoyed Science Time with Michael.